Hello, everyone. Welcome to our discussion on human genetics. In this session, we are going to discuss about gene mutation disorders. In gene mutation disorders, we'll be discussing on with autosomal gene mutations and inborn errors of metabolism. Autosomal gene mutations are those mutations or gene mutations that is affecting the autosomal genes, and we'll be taking two examples. They are brachydactyly and sickle cell anemia. Brachydactyly belong to the type of gene mutations or gene mutation disorder which is inherited in a autosomal dominant manner. And sickle cell anemia is a disorder or a condition which is inherited in a autosomal recessive manner. And the next topics will be the inborn errors of metabolism. So these also are gene mutation disorders where the mutation is causing a block in the metabolic pathway and will result in the particular type of disease disorders like L-captonuria, phenylketonuria, and albinism. So first we'll move on to the gene mutation disorders on that the autosomal gene mutations. The first one will be brachydactyly. So as we mentioned earlier, this is an due to an autosomal dominant mutation, and in the main feature of this particular condition is abnormally short fingers and toes. As it is due to a dominant gene, individuals who are both homozygous as well as heterozygous expresses this phenotype. The people who are having this condition can live their normal productive life, and they will be having difficulties, just the difficulties faced due to this short fingers and toes. Other than that, all the other phenotypic and the genotypic metabolism, etc., everything will be normal. And because of brachydactyly or brachydactyly occurs due to the abnormal development of phalanges as well as metacarpals or both. There are some more examples of autosomal dominant mutations. They are, the first one is polydactyly, which is the presence of extra digits. The second one is achondroplasia, which is a form of dwarfism. The third one is tylosis, which is excessively thick skin on the palms and toes. And the next one is anonesia, which is the absence of nails in fingers and toes, and dentinogenesis imperfecta, which is the crowns of teeth which wear down quickly. So these, along with the brachydactyly, are examples of autosomal dominant gene mutation disorders. Coming to the next one, which is sickle cell anemia. So sickle cell anemia is an autosomal recessive mutation, and hence only the individuals who are homozygous for this particular gene uh, will be will be expressing this particular condition. For individuals to be sickle cell anemia or sickle to have a sickle cell disorder should inherit the mutant gene from both the parents, that is from the father as well as the mother. So this particular disease is common in the places like Africa, the American blacks, as well as the tribes of Wayanad. So you can see what is the sickle cell mean. So in sickle cell anemia, you can see that this is a normal red blood cell, which is a normal RBC, which is a disc-like biconcave structure, which is highly flexible, and it does not stick to the other RBC because of the presence of empty number of uh, carbohydrates like sialic acids, which impart a negative charge. As a result, the other uh, RBC, which is also having a negative charge, repel each other as they do not. As a result, they do not clump. But when the mutation occurs in the particular gene uh, causing sickle cell anemia, what happens is that the shape changes to sickle cell. So we'll see this particular video about sickle cell anemia, what it is, etc.
In people with sickle cell disease, their red blood cells become deformed, turning
You can see the
sickle-shaped.
deformity now. So that
The
is the
bone
sickle
marrow
cell one.
tries to make more red cells to make up for the loss, but can't keep up,
You can
causing
see in the
anemia.
bone marrow, they are the normal discoid shape.
Their new shape keeps them from moving properly through the body, and the misshapen cells can jam up and stick to the walls of the blood vessels.
You can see that they're clumping together, which does not
These
happen
clumps
in
cut
normal
off
blood
oxygen
cells.
to healthy tissue, delaying a child's normal growth and causing fatigue and extreme pain.
So that was a short view of what this sickle cell anemia is.
In people with
So coming to the details of the sickle cell anemia. So RBCs are sickle shaped than usual biconcave disc, as we saw the particular picture and also the video. The normal RBCs move very easily through the blood vessels and capillaries because they are very flexible and because of the presence of certain special proteins on their membrane which help them to be to bind, to squeeze through the capillaries, etc. Mature RBCs are usually stuffed with a protein or a metalloprotein, which is called as hemoglobin, which is the oxygen carrying molecule or the oxygen carrying molecule in the RBC. So hemoglobin, coming to the structure of hemoglobin, as you see in this diagram, it consists of four heme groups. These blue dots, which you see here, which is enlarged here, this is one heme group which is having a porphyrin ring with an iron containing porphyrin ring, and we can see four protein chains. So such structures, you have such four structures here, and these four structures is capable of binding to four oxygen molecule. One can bind to one oxygen molecule, thus four can bind to four oxygen. So here what happens is the heme. So this is the structure of the heme, which is an iron containing porphyrin ring. And you can also, in the hemoglobin, there are four protein chains, which are two alpha, which is in red, which is shown here, and two beta, which is shown in orange. And the alpha contains about 141 amino acid and beta contains about 147 amino acids. And three types of hemoglobin is usually distinguished in humans. The one is seen in the adult humans, which is called as hemoglobin A. The other one, which is a fetal hemoglobin, which is hemoglobin S. The other one is the abnormal hemoglobin, which is generated as a result of a mutation, and that is called as HbS, which is S stands for sickle cell anemia. So sickle cell anemia is due to the abnormal type of hemoglobin in the RBC, which is called as HbS. And this HbS or this mutant hemoglobin is responsible for the change in the shape of the RBC, and this happens when the RBCs are exposed to low oxygen level. That is why in the video which we saw in the last slide, you saw that the bone marrow is making the RBC, which is of normal biconcave shape, and from the bone marrow, it reaches the blood, it reaches the lungs, where oxygenation takes place, and when it reaches the tissues, where the oxygen concentration is very less. Whenever these HbS containing RBCs is exposed to a environment which is very less in oxygen, it changes its shape to sickle. Okay, so that is what we saw in that particular video. When it reaches the organs or near the organs in the capillaries, etc., whenever there is a decrease in oxygen concentration, they will change their shape. The sickle cell RBCs are stiff and sticky, which is opposite to the normal RBCs, which is very flexible and it is non-sticky because normal RBCs repel from each other because of the presence of the heavy negative charges on them, which is imparted by the sialic acid which is present on the surface. And this sticky and stiff sickle cells, they tend to block the blood vessels of the limbs and the organs, which causes pain, serious infections, and also organ damage. So these individuals or this particular mutation is inherited in the short arm of the which is a gene which is located in the short arm of chromosome number 11, which codes for the beta protein or the beta subunit of the hemoglobin. there is a single base change or a single nitrogen base mutation at the sixth position of the amino acid. The sixth amino acid is replaced. In the normal beta hemoglobin in HbA, the sixth position, the amino acid is glutamate or glutamic acid. It is an acidic amino acid, and when a single base change occurs, this is replaced by, or this is coded by, or the replaced or the mutant codon will code for another amino acid which is called for valine, and this valine is a hydrophobic amino acid, and because of this substitution, a acidic amino acid is replaced by a uh, hydrophobic amino acid, and this causes a single change, a single amino acid of the 147 amino acid present. There is the change of only one amino acid, and that is resulting in this devastating change in the shape of the RBC. So it is in the gene is present in chromosome number one, and the change in the gene causes the replacement of glutamate to valine. So this is what happens in sickle cell anemia. So we'll watch this video.
What shape are your cells? Squishy cylinders? Jagged zigzags? You probably don't think much about the bodies of these building blocks, but at the microscopic level, small changes can have huge
So
consequences.
this is a
And
change
while
that is
some
happening to the
adaptations
RBC.
change these shapes for the better, others can spark
It
a cascade of debilitating
attains a sickle shape
complications.
like this.
This is the story of sickle cell disease. Sickle cell disease affects the red blood cells, which transport oxygen from the lungs to all the tissues in the body. To perform this vital task, red blood cells are filled with hemoglobin proteins to carry oxygen
So
molecules.
this is a hemoglobin
These
protein.
proteins float independently
You can see
inside
that
the red
this
blood
is what
cells'
is, we
pliable
said that it is
donut-like
stuffed
shape,
with
keeping
hemoglobin.
the cells flexible enough to accommodate even the tiniest of blood vessels. But in sickle cell disease, a single genetic mutation alters the structure of hemoglobin. After releasing oxygen to tissues, these mutated proteins lock together into rigid rows. Rods of hemoglobin cause the cell to deform into a long, pointed sickle. These red blood cells are harder and stickier and no longer flow smoothly through the blood vessels. Sickled cells snag and pile up,
You can see they are
sometimes
piling up
blocking the vessel
the
completely.
blood vessels.
This keeps oxygen from reaching a variety of cells, causing the wide range of symptoms experienced by people with sickle cell disease. Starting when they're less than a year old, patients suffer from repeated episodes of stabbing pain in oxygen-starved tissues. The location of the clogged vessel determines the specific symptoms experienced. A blockage in the spleen, part of the immune system, puts patients at risk for dangerous infections. A pileup in the lungs can produce fevers and difficulty breathing. A clog near the eye can cause vision problems and retinal detachment. And if the obstructed vessels supply the brain, the patient could even suffer a stroke. Worse still, sickled red blood cells also don't survive very long, just 10 or 20 days, versus a healthy cell's 4 months. This short lifespan means that patients live with a constantly depleted supply of red blood cells, a condition called sickle cell anemia. Perhaps what's most surprising about this malignant mutation is that it originally evolved as a beneficial adaptation.
So scientists believe that this particular mutation in the hemoglobin is a beneficial mutation. And why this is called as a beneficial mutation? Because this particular mutation was traced to be evolved in the regions where malaria was affected. The disease, which is called as malaria, was affected. The malarial parasite, in a stage of its life cycle, it is residing in the RBCs. So what happens is that this particular mutation causes the change in the shape, as well as it leads to, or it does not favor the particular individuals with the sickle shape will not favor the harboring of this malarial parasite. So scientists believe that this might have been evolved as a mechanism to tide over the malarial parasite. But what happened is that in such individuals who are heterozygous for this condition. We said that sickle cell anemia becomes, or it is a homozygous recessive condition. There are individuals which are heterozygous for the condition. So those individuals which are heterozygous for the condition will live normally. Normally, in the sense that in such individuals, half or so, if a particular person is producing 1000 RBC from the bone marrow, after 1000 RBC, what happens is 500 will be normal because half will be from one of the allele and half will be from the mutant gene because the person is heterozygous. So such heterozygous individual will have half HBS. That means whenever it is a low oxygen concentration comes, half will form the shape of sickle cell anemia. Half will be carrying out the process of oxygen transport. So when you see from the point of view of the malarial parasite, it can infect that particular RBC only half. Only half of the RBCs could be could be infected for harboring this particular malarial parasite. As a result, such individuals are at a very low risk for malaria. So this was evolved as an adaptation, but it became a particular condition or a life-threatening condition when it was evolved as a homozygous uh, recessive condition. That means if it was only in the heterozygous condition, it was normal because it was as an adaptation for thriving from malaria. But at a particular stage, when two heterozygous individuals gave birth to offsprings that resulted in individuals who are homozygous recessive, which results in a devastating condition, which is called as a sickle cell disorder. So that is what they are.
Researchers have been able to trace the origins of the sickle cell mutation to regions historically ravaged by a tropical disease called malaria. Spread by a parasite found in local mosquitoes, malaria uses red blood cells as incubators to spread quickly and lethally through the bloodstream. However, the same structural changes that turn red blood cells into roadblocks also make them more resistant to malaria. And if a child inherits a copy of the mutation from only
That is
one
what is
parent,
called as
there will be
heterozygous.
just enough abnormal hemoglobin to make life difficult for the malaria parasite, while most of their red blood cells retain their normal shape and function. In regions rife with this parasite, sickle cell mutation offered a serious evolutionary advantage. But as the adaptation flourished, it became clear that inheriting the mutation from both parents resulted in sickle cell anemia. Today, most people with sickle cell disease can trace their ancestry to a country where malaria is endemic. And this mutation still plays a key role in Africa, where more than 90% of malaria infections occur worldwide. Fortunately, as this adaptation thrives, our treatment for sickle cell continues to improve. For years, hydroxyurea was the only medication available to reduce the amount of sickling, blunting symptoms, and increasing life expectancy. Bone marrow transplantations offer a curative measure, but these procedures are complicated and often inaccessible. But promising new medications are intervening in novel ways, like keeping oxygen bonded to hemoglobin to prevent sickling, or reducing the stickiness of sickled cells. And the ability to edit DNA has raised the possibility of enabling stem cells to produce normal hemoglobin. As these tools become available in the areas most affected by malaria and sickle cell disease, we can improve the quality of life for more patients with this adverse adaptation.
So with this, we come to the end of this particular discussion of our sex chromosomal abnormalities. So we come to the end of our discussion with the the autosomal gene mutation abnormalities like brachydactyly and sickle cell anemia. In the next video, we will be discussing in detail about the inborn errors of metabolism, which is L-captonuria, phenylketonuria, and albinism. Thanks for hearing.